the next step in my experiment was gel electrophoresis. Now, gel electrophoresis is basically it's just another machine, and you make what's called an agarose gel. It's a lot like Jell-O in consistency. And you put your agarose gel into a tank, kind of like this, and then you pour it with buffer solution, and you load it with your final PCR reaction. Alright, the gel itself is prepared with ethidium bromide. So that just, ethidium bromide is just an intercalating agent, which means it combines itself with your DNA. That way when you look at your product, your final agarose gel product, under an ultraviolet light, you're going to see these fluorescent bands, which I'll show you in a minute. So after your gel is loaded and you pour the buffer on, you're going to expose it to voltage. Now, since there's a positive charge at one end and a negative charge at the other end of your machine, and we know that DNA has a slightly negative charge, it's going to be attracted to the positive charge on your electrophoresis machine. So the DNA is going to, run towards your positive. Now, one point I'd like to make about this is your DNA separates based on size. So in your gel, when you're loading it, at the first row, sometimes in the middle, and then at your last, you're going to have what's called a molecular weight marker. Now all that means is, if you think about it, you set up a racing course, for example, and you've got a nice shiny red Porsche with black racing stripes, and you've got a dusty old yellow school bus. All right, you put them both at the start line, and you've got your racing flag. And you go on your mark, get set, go! All right, who's going to get to the finish line first? Probably going to be the Porsche, right? So, in reference to DNA, the lighter the faster, the lighter the molecule, the smaller the molecule, the faster it's going to go through your gel. That's how it separates based on size. Smaller molecules move faster than larger molecules. So this is an example of your final product exposed to ultraviolet light. For this gel, I used what's called an ITS primer, which basically means it's a highly conserved primer. So if I had, way back when I did my DNA extraction, if I had any DNA at all in my tube, these ITS primers are going to pick that up and say, yes, you have DNA in your tube, so you can continue with your experiment. And we know this because we've got discrete, extremely bright bands, and they're all at the same place in the gel, right? Just below 250 base pairs. So I was able to move on to the next step. And what I was kind of looking for in my experiment was, how does ozone affect the environment as a whole? To reiterate, in the lab we expose something to ozone, and whatever that is, it gets decimated. It's gone. It's killed. We don't have to worry about it anymore. Is that true in the environment? We thought it was going to be. We thought the answer was going to be yes. We thought after exposure to ozone, we weren't going to find anything. Clearly not the case. These two bright bands right here, those came from samples that were exposed to ozone in the subsurface environment. So we were shocked. We couldn't believe it. This was kind of a groundbreaking experience for us because, when we set up our experiment, we thought it was going to turn out one way when that clearly was not the case. This gel right here is just a picture of a typical gel that I got, as a product of, my PCR reaction and gel electrophoresis. We've got polymorphic amplification and specific binding. So with polymorphic amplification, we can see here in this lane, for example, I've got 3 different gels, or 3 different bands in my gel, okay? And that just means that maybe during my PCR reaction, there was an A instead of a T or something like that, so, so you see more product, all right? And we can see specific binding because we've got discrete bright bands in certain lanes. So, you know, that's basically what I saw in my experiment.